This is the sequencing system that we use. Here's an Illumina iSeq 100 cell that basically will be plugged in with its cartridge. And on this chip type system is a flow cell, a little football shaped area there, where basically about, it's probably about 7 million micro pits made into a surface there, each one filled with some oligos to capture one strand of DNA where they're replicated for simultaneous sequencing. All the chemistry's on board this cartridge. It'll be used once. This one was used once. And it's probably about $500 to $600 for the cartridge and its flow cell. It's used once, but if you sequence 25 or 50 patient samples on it, you get the whole cost down with the reagents to make the sequencing library. You're down to about maybe $250 to $300 to sequence 8 genes for somebody that might have a disease caused by a variant in one of those genes. This is the little system that does the sequencing. It will sequence 1.9 billion bases of DNA over about 18 hours. It's one of their small sequencers, only does 1.9 billion bases. And we'll see if it will unload its door for us here. There you go. It's kind of like Star Trek, eh? And so normally this cartridge, there's a little foil cover here that we break 20 microliters of a combined library samples put into there, just 20 microliters. This goes into that rack and then basically is loaded into the sequencer to run under the parameters that we set. So this has an onboard operating system. It's got a Windows interface. It's actually, the computer in here actually has an Apache web server because the software, you talk to it using your web browser to a web server that's right in the instrument. This will align all the sequencing variants for all the patient's files and tell us what the differences are. All right there on your desktop.